Hi guys, today's topic is plant breeding, single cell proteins and tissue culture. Let's learn about plant breeding. What is plant breeding? It is the purposeful manipulation of plant species in order to create desired plant types which are better suited for cultivation, give better yields and also disease resistant. Today, all our major food crops are derived from domesticated varieties. Plant breeding can broadly be classified in two things: classical plant breeding and modern plant breeding. Classical plant breeding includes crossing of superior pure line and selection of plants with desired characteristics. Modern plant breeding is nothing but the desirable plant trait wished to be incorporated by the plant breeding. The desirable plant trait includes increased crop yield, improved quality, tolerance to environmental stresses, pathogen resistance, tolerance to insects and pests. Let's learn about the steps involved in breeding a new genetic variety of a crop. The first step is collection of genetic variability. Genetic variability is availed from the wild relatives of the crop. Here, all the wild varieties and the relatives of the crop are collected and preserved. The natural genes available in the population are utilized by this method. The entire collection of plants which are wild types or relatives of a given crop which has all the diverse alleles for all genes is called as germ plasm collection. The second step involves the evaluation and selection of parents. For the available genetic variability, the germ plasm is analyzed and evaluated to identify the plants with desirable traits. The third step involves the crop hybridization among selected plants. Two selected plants are crossed which are nothing but hybridized. These facilitates the combination of desired traits from two different plants. Pollen grains from one plant are dusted over the stigma of the other plant. The fourth step involves the selection of superior recombinants. Among the hybrid progeny, those plants are selected which have the desired character combinations. Careful scientific evaluation of progeny is required for selection. This step yields the plant that is superior to both the plants. The fifth step involves the testing, release and commercialization. Selected yields are evaluated for traits like quality, disease resistance, insect resistance, etc. Let's learn about the Indian hybrid crops. Firstly, the wheat and the rice. In 1960s, wheat and rice production increased tremendously. Norman E. Borlaug developed a semi-dwarf variety of the wheat. Sonalika and Kalyan Sona are the two of the hybrid wheat varieties which are grown in India. Semi-dwarf wheat varieties were taken from IR8, which is nothing but International Rice Research Institute, which is situated in Philippines, and Taichung Native 1, which is situated in Taiwan. Jaya and Ratna are the better yielding semi-dwarf rice varieties that were later developed. Another Indian hybrid crop is sugarcane. Saccharum barberi is a native sugarcane species present in North India, whereas Saccharum officinarium belongs to South India. Saccharum officinarium has thicker stems and high sugar content, but it does not grow well in North India. These two varieties were crossed to get the desirable characteristics or the qualities of both, which includes higher sugar content, thicker stems, and the ability to grow in North India. Let's learn about another Indian hybrid crop called millets. Hybridized maize, jowar, and bajra have been successfully developed in India. These varieties have high yielding and resistant to water stress. Now, applications of plant breeding. If the resistance to a particular disease is already present in a plant, it reduces the dependence of the plant on fungicides and bactericides. Before breeding, one must know the causative agent of a disease and its mode of transmission. Some common diseases like the fungal disease, that is brown rust in wheat, red rot in sugarcane, and late blight in potatoes. The bacterial diseases include the black rot in crucifers, whereas the viral disease is the tobacco mosaic virus. Disease resistance can be provided by three methods: the conventional breeding, the mutational breeding, or the genetic engineering. Let's learn about the conventional breeding. It includes the basic steps of screening, germplasm, hybridization, selection, testing, and release. For example, Himagiri, a wheat variety which is resistant to a leaf, stripe rust, and hill bunt, whereas the Brassica variety, that is Pusa swarnim, which is resistant to white rust, are bred by conventional breeding. One limitation to this method is that the genes for disease resistance are limited in number. Let's learn about the mutational breeding. In this method, genetic variations are created, and as a result, the creation of traits are not found in the parental type. Mutations are induced with the help of mutagens, that is nothing but the chemicals or irradiation. The plant in which the desired character, that is nothing but the resistance, has come through mutation which are selected. Now let's learn about genetic engineering. Certain wild varieties such as which has disease resistant characteristics but they are low yielding. Disease resistant gene from such varieties are introduced in high yielding varieties through a technology called as recombinant DNA technology. For example, in mung bean, resistance to yellow mosaic virus and powdery meadow were induced by mutations. The second example could be the resistance to the yellow mosaic virus in bindi. The scientific name for bindi is Abelmoschus esculentus, was transferred from the wild species to produce a new disease resistant variety of bindi which is called as Parbani Kranti. Let's learn about the pest resistant crops. Certain morphological characters like hairy leaves, solid stems of wheat which are naturally provide resistance from insects and pests. Similarly, biochemical characters provide resistance from insects and pests. For example, the high aspartic acid, low nitrogen and sugar content in maize leads to resistance against the maize stem borers. Such varieties are bred with non-resistant varieties to produce pest-resistant hybrids. For example, Pusa Gaurav variety of rapeseed mustard, which is nothing but Brassica, is resistant to aphids. Pusa Sem 2 and 3 of flat bean is resistant to jacids, aphids and fruit borers, whereas Pusa Savani and Pusa A4 of bindi is resistant to shoot and fruit borers. Now let's learn how to improve the food quality. A large number of people around all over the world suffer from micronutrient, protein and vitamin deficiencies since they cannot afford to buy food rich in these nutrients. Such deficiencies lead to diseases, mental retardation, reduced lifespan. An alternative to this problem is to breed crops that are rich in nutrients. Such approach is called as biofortification. The objectives of biofortifications are to improve protein content and quality, oil content and quality, vitamin content, the micronutrient and the mineral content. For example, the maize hybrids which are developed in 2000s have twice the amount of lysine and tryptophan amino acids compared to other maize hybrids. Another example could be Atlas 66, a wheat variety having higher protein content. Now let's learn about single cell protein called as SCPs. Conventional agricultural production of cereals, pulses, vegetables, fruits, etc. may not be able to meet the demand of foods at the rate at which human and animal production is increasing. The shift from grain to meat diets also creates More demand for cereals as it takes around 3 to 10 kgs of grain to produce 1 kg of meat by animal farming. More than 25% of the human population is suffering from hunger and malnutrition. One of the best alternate sources of proteins for animal and human nutrition is single cell proteins. Single cell protein means that the microbes are used as a source of proteins. Microorganisms, despite being very small, are capable of producing tons of proteins due to their high rates of biomass production. Just like mushroom culture, it is expected that the microbes will soon be accepted as the source of food. Presently, spirulina, an algae, is widely accepted as the source of single cell protein. It is economical and eco-friendly as well. It can be grown on economic substrates like the wastewater from potato processing plant, the straw, the molasses, or even sewage to produce large quantities and can serve as food rich in proteins, minerals, fats, carbohydrates, and vitamins. Incidentally, such utilization also reduces environmental pollution. It is calculated that 250 kgs of cow produces 200 grams of protein per day. In the same period, 250 grams of microorganism like Methylophilus methylotropus, because of its high rate of biomass production and growth, can be expected to produce 25 tons of protein. Now, let's learn about the tissue culture. Tissue culture is the process of developing a complete plant from a part of a plant, which is called as explant. Explants can be grown in sterile conditions in special nutrient media to regenerate the complete plant. Nutrient media could contain a carbon source, which could be sucrose, organic salts, vitamins, amino acids, and phytohormones. Hence, here the propagation is achieved for a large number of plants in a short duration. This process is called micropropagation. All the plants which are obtained through tissue culture are called as soma clones, since they are genetically identical to each other as well as the parent plant. There is another process which is used in tissue culture called as somatic hybridization. It refers to the hybridization of a somatic parts of two different plants. Protoplasts are isolated and fused to get a hybrid protoplast which grows further to form a new plant. The new plant formed is called as somatic hybrid. For example, the protoplasts of the potato and the tomato have been actually fused to form tomato, but this plant is not commercially viable. Let's learn about the application of tissue culture. Many important plants like apple, banana and tomato have been grown on laboratory scale by using this method. This method can be used to recover a healthy plant from a diseased plant. For example, a plant inflicted with virus may not have virus in its apical or axillary meristems. Hence, these parts can be cultivated to obtain a healthy plant. Thank you.